Hello friends, I am Gaurang Makwana. I welcome you all to my another video tutorial Biotech guide. In this video lecture, we are going to discuss about a post-translation modification, where we are going to discuss main 5 important types of a post-translation modification based on addition of a chemical group, complex group, then polypeptides, cleavage of a protein, and modification of amino acid. Then also at the end, we are going to brief about this all post-translation modification and their function. So as usual, today's video going on a very interesting level. Stay tuned with us and enjoy this video lecture. Let us start first with introduction. What is post-translation modification? So as we discussed in our previous video, post-transcription modification, RNA need to be modified or stable via processes known as a post-transcription modification, where polyadenylated tail is attached and capping process is also there, and another RNA splicing take place. So these are some steps which are required in the post-transcription modification. Here it's same post-translation modification. After synthesis of a protein need to be modified for performing a various cellular function. So here post-translation modification is a process where protein structure and function are altered after the translation by the addition or removal of a chemical groups to specific amino acid residue. So this is the simplest definition of a post-translation modification. Now let us discuss types of a post-translation modification. So basically, based on the addition of groups or some processes, post-translation modification is classified into the 5 types. First is based on the addition of a chemical group. Again, 4 subtypes are there: phosphorylation, acetylation, hydroxylation, and the methylation. Then second type, based on the addition of a complex group. Again 3 subtypes: glycosylation, ampicylation, lipidation. Then third, addition of a polypeptides. 1 subtype is there, ubiquitination. Then a process, cleavage of a protein is also one kind of a post-translation modification, and the subtype is proteolysis. And at the end, amino acid modification, deamination is the subtypes of amino acid modification types of a post-translation modification. So in this video lecture, we are going to brief about all these types. Let us start quickly. First, based on addition of chemical group. First subtype is a phosphorylation. So here I am going to tell about one important point, that is we are going to memorize using a mechanism, then enzyme which is used for this mechanism, and the third application. So these 3 main important points you have to write in your exam as well as any kind of a theory. So first, phosphorylation. So which mechanism are used here? Phosphorylation and dephosphorylation. So phosphorylation, that means addition of a phosphoric group, and dephosphorylation, that means a removal of a phosphate group. So here phosphorylation take place in which kind of amino acid? Serine, threonine, or a tyrosine amino acid residue. Okay, so this is the mechanism. And here one important point that you have to memorize, that we know protein is made up of a amino acid. So here any kind of modification process is happen on amino acid residue. Okay. Then another second important point is which enzyme. So here 2 enzymes take place, kinase for the phosphorylation and phosphatase for the dephosphorylation process. Then third, application. So basically phosphorylation have several implications like cellular processes including cell cycle, growth, apoptosis, and a signal transduction pathways. So these are the application. Now another 2 points are is effect. So after addition of a phosphate, what happen? As we know, one of the key important example is DNA, addition of DNA having a phosphate group, okay, so they having a basically negatively charged. So here addition of a phosphate group can convert a previously uncharged pocket of a protein into a negatively charged and a hydrophilic protein, thereby inducing a conformational changes in the protein. So basically phosphorylation resulted into the hydrophilic and negatively charged protein. Then another second point is example. So basically activation of a p53 tumor suppressor protein which is used in a cancer therapeutics and activated by a phosphorylation. It's N-terminal and several kinases enzymes. So this is the one important example. And several enzymes or signaling proteins are switched on and off phosphorylation or dephosphorylation. So this is also one important point of a phosphorylation. Any mechanism start one kind of a switching mechanism is performed via this phosphorylation process, and this is the basic reaction where the protein is phosphorylated. So here ATP is required for the phosphorylation. By the use of a kinase enzyme, protein is converted into the phosphoprotein and ATP is converted into the ADP. So this is the first our phosphorylation. Second, acetylation. So here which kind of mechanism is there? Addition of a acetyl group. Okay, so this is the mechanism, and which enzyme is responsible for acetylation? So here acetyl transferase is enzyme for the mechanism of acetylation. So here in this post-translational modification, specifically lysine acetylation, the enzyme is often called as a lysine acetyl transferase take place, and mostly 90% of a human protein undergoes this kind of a process, and acetylation. Okay, so here for the amino acid specific lysine acetyl transferase enzyme take place. Then application. So basically acetylation process take place in several biological function, including protein stability, location synthesis and apoptosis, also cancer and DNA stability. So these are the application of this process. Another important point is acetylation and deacetylation of a histone form a critical part of a gene regulation. So this is the main important point. As we know, DNA having a negatively charged, okay, and acetylation provide basically negatively charged. So here we know that histone is a positively charged. That's why DNA is rolling around to histone protein and make a compact structure. But here for the any kind of a process, DNA will be unwound via acetylation process. How? So here addition of acetyl group provide negatively charged to a histone protein. So negative repulsion is occur, and in result basically unwinding of a DNA take place. So this is the one important point of a acetylation process, and that will be accessible for a gene transcription or for the translation process. Then acetylation of a p53 tumor suppressor gene is crucial for its growth and suppressing properties. So that is also mainly performed via acetylation process. So these are the some important points for acetylation process. Then third subtype in addition of a chemical group, which is hydroxylation. So which mechanism? Addition of a hydroxyl group. Then which enzyme? Hydroxylase enzyme. And application, so converting hydrophobic or lipophilic compound into hydrophilic compound. So this is hydroxylation. And the last, in addition of a chemical group, which is a methylation. So mechanism is methylation, addition of a methyl group, okay, in which amino acid residue, lysine or arginine residue of a protein. Then one important point here, arginine can be methylated one or twice, while lysine can be methylated one, twice or thrice. Then which enzyme? Methyl transferase enzyme. And application, so basically methylation provide this kind of gene activation or repression based on the residue, and also folding activity. So these are the some important application which is performed via methylation process. Here we have to read that methylation has been widely studied in a histone, wherein histone methylation can be lead to gene activation or a repression based on a residue that is methylated. Also influence the protein folding by increasing the hydrophobic nature of amino acids. So these are the application which gene activation, repression, and also protein folding. And this is the simplest mechanism where S-adenosyl methionine is converted into the S-adenosyl homocysteine via methyl transferase enzyme. So this is all about of a addition of a chemical group post-translational modification. Then second, based on addition of a complex group. First subtype is a glycosylation. So here which mechanism? Addition of a oligosaccharide, also termed as a glycan. Okay. Two terms included here, N-linked glycosylation and O-linked glycosylation. So basically in amino acid, amino group is also there and hydroxyl group is also there. So when glycan is added at the N terminal side is defining as a N-linked, and where any glycan is attached with oxygen atom is defining as a O-linked glycosylation process. Here N-linked glycosylation occurs in the amide nitrogen of aspargine, while O-linked glycosylation occurs in an oxygen atom of a serine and a threonine. So these are the some conditions which required for the glycosylation process. Then which enzyme take place here? Glycosyl transferase enzyme take place for addition of a sugar residue to a protein during glycosylation. Then what application are there? So protein sorting, immune recognition, receptor binding, inflammation, and pathogenicity are the application for glycosylation. Then here one example is there. N-linked glycan on an immune cell can detect how it migrates to specific sites. Similarly, it can also determine how a cell recognizes self and non-self. So this is the one key example of a glycosylation process. Here 4 classes are included, as I discussed. When any glycan is attached at the site of a amido group, that is defining as a N-linked, oxygen group is defining as a O-linked, and similar like that. So here N-linked glycan example are aspargine and arginine. O-linked glycans examples are serine and threonine. And when in combination of a phospho and oligosaccharides, that is defining as a phosphoglycan, is linked through a phosphate of a serine, and this is the rare form of a sugar added to a carbon or a tryptophan side chain is defining as a C-linked glycan. So this is the glycosylation. Second, amylation process in a addition of a complex group. So here which mechanism is there? Addition of a AMP, adenosine monophosphate molecule, to the protein, and this process is defining as an amylation process. It involves the covalent attachment of a AMP molecule to a specific amino acid side chain to the protein and acting as a post-translational modification that can alter the protein function and activity. Then which enzyme take place here? Adenyl transferases, and also defining as a amylases. So these are the enzyme here take place for this process. And application, so amylation is used as a regulatory mechanism impacting protein interaction, enzyme activity, and cellular signaling pathways. So these are the application. Important point is this process is reversible. Where addition of a AMP to a protein, it involves a formation of a phosphodiester bond between the hydroxyl group of a protein and a phosphate group of a AMP. So this process is also can be done by reversible. So this is the amylation. Then last in addition of a complex group, that is a lipidation. So here basically three different significant process take place. Addition of a lipid group, that is defining as a lipidation process. So here that is subdivided into a prenylation and myristylation, palmiolation, and a glycosyl phosphatidyl linositol GPI anchor addition. So these are the some important significant process in lipidation. First, prenylation mechanism: addition of a isoprenoid moiety to cysteine residue using a acyl transferase enzyme, and plays a critical in controlling the localization and activity of several protein. Have a crucial function in a biological regulation. So this is the prenylation. Then myristylation involves the addition of a myristoyl group to a glycine residue by an amide bond, and it has an application in membrane association and apoptosis. Then in third, palmitylation, our palmitoyl group is added to a cysteine residue of a protein. Then last important one, GPI anchor addition, defining as a glycosyl phosphatidyl linositol GPI. Anchor addition: the carboxyl terminal signal peptide of the protein is split and replaced by a GPI anchor. So this is the mechanism, and this is important because recent research in human genetics has revealed that GPI anchors are important for human health. Any defects in the assembling, attachment, or remodeling of a GPI anchors lead to a genetic disease known as a inherited GPI deficiency. So this is the second, lipidation. Then third, based on the addition of a polypeptide. Here polypeptides, that means a small molecule of a protein which is defining as a ubiquitin. So let us see ubiquitination process. Mechanism: ubiquitination involves addition of a protein found ubiquitously termed as a ubiquitin to a lysine residue of a substrate. If a single ubiquitin molecule is there, so defining as a monoubiquitination mechanism, and if several, so defining as a polyubiquitination. So this is the mechanism. Then enzyme: so here ubiquitinate ligase enzyme take place, and application: so here ubiquitination helps in signaling pathway, inflammatory process, DNA process where monoubiquitinated protein may influence cell tracking and endocytosis. So here this is the ubiquitination process. One important point is polyubiquitinated protein are recognized by 26 subunit of a proteasome and are subsequently targeted for a proteolysis and the degradation. Here simplest mechanism is defining is there: protein is ubiquitinated by a ubiquitinate ligase and converted into the ubiquitinated protein, then proteasome and the peptides. Then fourth, based on a cleavage of a protein. So here mechanism: proteolysis, that means a breakdown of a proteins into a smaller polypeptides or amino acid. Then which enzyme? Protease enzyme take place, and function is for activation. So here one example is there: inactive protein, that is pepsinogen, is activated via protease and converted into a pepsin, active form of a enzyme or a protein. Here removal of a N-terminal methionine, a signal peptide, after translation lead to be conversion of an inactive or a non-functional protein to the active one. So this is the another example. Then last, based on amino acid modification, deamidation process is there, where mechanism is there but enzymes are not defined. So here deamidation is the process of removal or a conversion of a asparagine or a glutamine residue to another functional group. Asparagine is converted to aspartic acid or a isoaspartic acid, while glutamine is converted into a glutamic acid or a pyroglutamic acid. This modification can change the protein structure, stability and function. So these are the application. So here we completed types of a post-translational modification. Then now let us quickly revise all function. First, phosphorylation activates enzyme like protein kinase A, regulates cell cycle progression involved in a signal transduction pathway. Then acetylation modifies histone protein to regulate gene expression, then can affect a protein-protein interaction. Then third, glycosylation adds sugar moieties to protein, impacting protein folding and cell surface recognition. Also important for cell adhesion and immune response. Then methylation can affect a protein DNA interaction, particularly in epigenetic regulation. Also modifies signaling pathways. Then last, ubiquitination target a protein for a degradation by the proteasome and plays a role in a cell cycle regulation and a stress response. So these are the example of a post-translational modification.